Well, thank you very much, and it's great to be at a meeting in person, I gotta say. So everyone's aware of the profound impact CAR T cells have had on hematological malignancies. But of course, 90% of all malignancies are from solid tumors. That is what we are focused on. So what I'll do is first go through all the various challenges, of course, of solid tumors, targeting those with CAR T cells, and how we're addressing all those challenges. So a big problem with all targeted therapies is on-target off-tumor toxicity. This is particularly the case with solid tumors where most antigens are tumor-associated. They're not tumor-specific. So you have some lower level of expression on non-cancer cells. With targeted therapies, you can simply reduce the dose. You can't do that easily with a live cell like a CAR T cell. When they see their target, they clonally expand, and you can have a lot of them present. That's not how you can get around the on-target off-tumor toxicities. Our affinity tuning does that, and I'll talk about that. Antigen loss is another major hurdle. I think that was brought up this morning. Again, that's also a problem with all targeted therapies. You target one antigen on a tumor, the tumor can lose that antigen, and it escapes. So again, indicate how we're focusing on that with our technology is a number of ways. T-cell exhaustion, that's clearly been an issue in all types of cellular therapies. The affinity tuning, what that does is essentially allows the T-cell to work more like a normal T-cell. Most T-cells interact with their targets at the micromolar affinity level. That's the normal interaction, forget T-cell killing. Most targeting moieties for CAR T cells are in the nanomolar. So this is one reason you get the exhaustion more in CAR T cells. So we think our affinity tuning addresses that. The tumor microenvironment, that's always a problem. So there are various inhibitory factors of the tumor microenvironment. I'll indicate how we're getting around that. And also compromised tumor cell trafficking. Obviously, that's an issue with solid tumors, and not so with hematological malignancies. So in terms of the again, what I'm going to stress is our focus is both on tuning the CAR T cells and be able to track them in the patient. So this is what I'll be talking about. And we believe that our tuned CAR T cells, as I indicated, address the on-target off-tumor toxicity. So by reducing the affinity, you can avoid interaction with basal cells having low level of expression. Antigen loss is another thing. There are two ways actually we're getting around that. One is we're going to be looking at dual targeting CAR T cells. The other thing, our first target, ICAM1, is actually upregulated in the presence of activated T cells. So with all tumor antigens, you don't get 100% of the tumor expressing the target. You'll get maybe 60%, 70% if you're lucky. What happens with ICAM1 is as soon as CAR T cells start attacking those cells, they secrete interferon gamma, which upregulates ICAM1 on the remaining cells. T-cell exhaustion, again, there are various ways of avoiding that. We believe the best way is to make the affinity more in the range of what T cells are normally accustomed to, which is the micromolar range, and this was really the basis of Affyimmune, this finding. Tumor microenvironment, various cytokines can help overcome that, both for preserving the function of the CAR T cells and also engaging other members of the immune system. So a variety of other cells, other T cells and NK cells can be activated by their cytokines. So this is something we're also looking at. And compromised T cell trafficking, again, if you have affinity tuned CAR T cells, they're also more able to traffic to the tumor. And I think our tracking, as I'll show you a little bit later, our tracking technology also addresses that. This is very briefly our pipeline. So we are currently in the clinic in a phase 1 study with targeting CAR T targeted ICAM1 tumors. In particularly, this is anaplastic refractory thyroid cancer. Very high levels of ICAM1 expression. We'll be expanding that indication early next year to other tumors such as gastric and triple negative that also have high ICAM1 expression. Beyond past that, again, we have others where, as I indicated, inducible cytokines to help overcome issues of the tumor microenvironment, and also dual CARs to address antigen escape. So what exactly is affinity tuning and how does that work? So you see on the left panel here, you can have various levels of antigen density in a cell. What we do is essentially tune the affinity such that if a cell has something on the order of 10 to the 5th antigenic targets on the surface, it will be recognized. If it has less than that, as is present in normal cells, it does not recognize those cells and will not kill them. So this is what our tuning does. It spares normal cells in addition to really opening up a variety of different tumor-associated antigens that we can target. In that, as I mentioned, there are almost none that are truly tumor-specific. In terms of tracking, this is done by adding which I'll mention briefly, a somatostatin type 2 receptor for which there's an approved radiolabeled tracer people can use for imaging neuroendocrine tumors that overexpress this receptor. What we're doing is simply expressing this same receptor in the CAR T cells, somatostatin type 2 receptor, such that when the patient is infused with the approved radiotracer, one can see exactly where in the patient the CAR T cells are. And what one sees here is simply a murine experiment where you see the classic example of initially where there's very low tumor burden, there's low CAR T cells. Once they recognize that there's tumors in the lungs, they expand. Once the tumor is attacked and killed, you can see that the immune response also resolves. And this is a classic immunological response, cellular immunological response. So this is now our whole platform, essentially, what we're developing. You can see down below the SSTR2, that's the somatostatin type 2 receptor. We're also generating CAR T cells that locally deliver cytokines that combat the tumor microenvironment. We also have dual CARs, as I mentioned, which we'll be generating, as well as other means of promoting the activity of the CAR T cells. So this is what, now I should also point out that for ICAM1, the targeting moiety of the CAR T cells is a portion of the natural receptor for ICAM1. This is LFA1. So fortuitously, this also interacts with murine ICAM1. So we can look at the toxicity in mice with this CAR T cell in addition to looking at tumor activity. So what we can see on the right panel in terms of the toxicity, if one has high affinity CAR T cells, mice will not survive, and there's actually toxicity, fatal toxicity because of those high affinity CAR T cells. If there are no CAR T cells or non-transduced CAR T cells, the mice succumb because of the cancer. But the green line indicates the affinity tuned CAR Ts get rid of the cancer and do not kill the mice. On the left is just an indication of various immunohistochemical stainings of various cancers showing the high level of expression of ICAM1 in tumor tissues relative to the expression in normal tissues. So in this example of a murine experiment, so we can see with the high affinity, again, as I indicated, one gets lethality to those mice because the CAR T cells recognize basal levels of ICAM1 expression in the mice. If you get kind of an intermediate affinity, you see an improvement in terms of both the anti-cancer activity compared to the non-transduced T cell group. But still, it's not optimal. The tumor cells come back. Whereas you have the optimally tuned CAR T cells, you can see you get much better efficacy in addition to safety. So this was really the finding, and this is micromolar affinity now. So this was really the kind of the pivotal observation that drove us to bring this compound into the clinic. So the first indication I mentioned was anaplastic refractory thyroid cancer. It's a rare disease. We have both orphan indication and fast track designation. We plan on expanding that to the more common tumors, triple negative breast cancer and gastric cancer. And on the right, you can see the activity in those animal models, that there's good activity of our compound ASC100 that's in the clinic against those types of tumor models. So this is very briefly our phase 1 trial that's currently ongoing. It's a modified 3+3 dose escalation, starting with 10 million, then 100 and 500 million CAR T cells dose, looking at cytokines and CAR T cells circulating in the blood. In addition, imaging is, of course, very important because that's part of our both way of monitoring the tumor, which is done in the clinic, in addition to tracking the CAR T cells, which we're able to do. Primary objective, of course, is safety. Secondarily, looking at the biological activity of the CAR T cells, both from any clinical observations as well as correlations between CAR T cell trafficking and the tumor response. So this is just what this looks like in one of the patients. So one gets a baseline scan early on, and there's always some background in this, and then followed out for several weeks after infusion. Again, this is a patient, one of our, in our earliest doses level, 10 million cells, comparing both FDG, which fluorodeoxyglucose that images the tumor and metastases, versus gallium-labeled dotatate, which images the CAR T cells because of their somatostatin receptor expression. So if you look at that first, just looking at the right-hand panel, this is now the average standard uptake values of both FDG and gallium-labeled dotatate in the patients after infusion. And we can see that after about 14 days, there's on average of 10 different lesions, a decrease in the level of FDG staining, which is concomitant with an increase of dotatate staining. And this correlates with the expansion of CAR T cells that we've observed in the blood after 14 days, which you see on the left-hand panel. Interesting in this patient, the patient had other therapies, radiation, also received pembrolizumab, and we can see after this an expansion, re-expansion of the CAR T cells, which has been noted previously in other studies. But it was interesting, and both these observations were gratifying to see in a patient that received such a low dose of CAR T cells. So just briefly to our candidates that are currently, that we plan to file IND on shortly. This is the AIC 1010, which now has cytokine secretion in addition to targeting ICAM1. And we can see on the right-hand side, this is now an intraperitoneal gastric cancer model, which is notoriously difficult to treat. We're seeing very good activity compared to the non-transfused T cells. This is another target, so this is a separate target now, epcam, that we're targeting. And one can see on the right essentially how this tuning works. So this is now a cytotoxicity assay with normal liver cells or normal kidney cells. And one can see with the green line, again, this is high affinity now, one gets significant lysis of normal cells with high affinity CAR T cells, whereas if you affinity tune that, reduce the level, you get essentially no toxicity in those normal cells, either normal liver cells or kidney cells, and you still have good anti-cancer activity, as you see on the left. So this is an example of how we're tuning the CAR T cells for optimal activity, both for the T cell and for avoiding on-tumor, on-target, off-tumor toxicity. So this is just the team. So the company was founded by Moonsoo Jin and Gang Song. They did their postdoctoral fellowships in Tim Springer's lab at Harvard, studying the interaction between ICAM1 and its natural ligand. And it was during the course of those studies that they identified the various affinity variants. And this really led to the founding of Affyimmune. They showed that you can do actually a structure-activity relationship with these affinity variants. That was the formation. The board of directors, we have also Simon Song, former head of Goldman Sachs in Hong Kong, Elaine Yang, and very happy to have as a scientific advisor Keith Knudsen, who has a long history in adoptive cellular therapy in addition to Cassian Yi, and of course Jim Millay, who has been really kind of a grandfather of immunology for many years, very respected in the field. So with that, I think I hope I've indicated that we have a pioneering platform that's addressing all those key challenges, the critical challenges for going after CAR T cells. We're a clinical stage company, so we're going to be expanding those trials from our initial orphan drug indication to more common cancers. And again, so we have multiple assets. As I showed you, we're able to take a variety of different cancers of our targets and tune the CAR T cells such that we can now target those targets effectively. And I think there are a lot of those. I mean, the story of targeted therapy is exactly that you have, on target, off tumor toxicities. So I think we're challenging that pretty effectively, and that's what you'll be seeing more from Affyimmune. So with that, I actually have about 20 seconds left. Happy to take any questions either now or afterwards. Thank you much.